Hi, this is Matthew Bickley, and I'm here to discuss my Hill Air Force Base Duck Pond microcosm. Quick little overview. Microcosms are small samples from environments. You get to see what life develops and how the organisms interact with each other. This gives ecologists portable ecosystems to observe and study and allow for observations that could not happen in the natural environment. I did two microcosms. One was made with artificial materials, so I built it myself, and then the other one was sampled from Hill Air Force Base Duck Pond. One thing to note is that after about week 3, I stopped taking notes on the artificial one just because I didn't think I was going to include this in the project, but I ended up doing it just because I thought it great. It made a pretty good control, kind of. Okay, so moving on. This is the actual Google Maps of where the duck pond was located. It's on Hill Air Force Base, like I said. These 3 ponds over here were completely iced over when I sampled it, and this was the only one that actually had open water. That red dot in the center is approximately where I took it. So I actually did this because I wanted to see if there would be insect life inside the pond where I was going, because I thought since everything was frozen over, they'd be dead. It'd be too cold for there to be invertebrate life into the pond. So I wanted to kind of test that out, and that's why I chose to do this area specifically. Um, this experiment it was over the course of 7 weeks for the artificial one and 5 weeks for the Hill microcosm. Moving forward, they're going to be referred to as the Hill microcosm and the artificial microcosm, just so that there's clarity and everyone understands which one I'm talking about specifically. So my hypothesis for the Hill microcosm was that the bottle will contain no heterotrophs due to samples being taken after a long cold front entering the area, and that everything was mostly frozen over. Like I said earlier, this was the only part that wasn't completely iced over. Even here, you can see ice, and on this side there's ice. This was the only open water that I had access to. And then the bottle will become overrun with algae due to the intense lighting conditions that'll be under it. I'll go into that a little bit later, but I didn't just set it on a windowsill. I had a lot more intense, more lighting than that. That's why I made that hypothesis. So the hypothesis for the artificial microcosm was that the snail eggs that I placed in it would not hatch due to the water quality and temperature, and that the algae would overtake the jar due to light exposure and zero contact maintenance. So methods and materials, site prep. I have 2 children and 2 dogs, so I really didn't want them moving it around and stirring up the substrate or heaven forbid breaking it and it gets everywhere. So what I did was I made it inside a cupboard. That's why I have this light panel right here, and that way it could shine light into it as well. So the ambient temperature inside this was 22 degrees Celsius, and this is on a heat pad for the artificial one, but I did make sure that it did not affect the Hill Air Force Base one. So it's just sitting in 22 degrees Celsius temperature. And then this is on an 8-hour timer. So it only goes off for 8 hours a day. That's how I know it's 8 hours of direct light. These don't look like it, but they are offset. So this one's a little further back than this one. So both have direct access to the light. Methods and materials for the Hill Air microcosm. So I have one mason jar. Its dimensions are height of 16.5 centimeters. Its length and width are both 8.6 centimeters. I have a shovel for collecting dirt. I have a measuring cup for measuring the dirt and the water collected. And then I have that light panel with the timer set for 8 hours of direct light, like I already stated. So methods and materials we have for the artificial microcosm. This is a complete cylinder jar with a clear latching lid. This is where it latches down, so it's clear, so you can actually see straight down it. I really like that about it. And then the heat pad placed on top is on the bottom of the tank, and it's got a light panel set and with 8 hours of direct light. It's gonna be in the same area as the other one. That's why that says there. So it's the same setup. This is the fish tank that I actually sampled the water from. This is where I got a lot of the materials from. I actually used clippings from the plants in here when I built it. So building the artificial microcosm, I used the water from that fish tank in the previous picture, and that way it already had a microbial substances inside of it, and it was already basically set up. I used 3 crushed floor tablets in the bottom of jars of like a fertilizer. I had 6 inches of biostratum aquatic substrate, and then I put 1 inch of play sand on top of that. I had 20 nearite snails on a rock, and then I threw a stick in there too, just for decor than anything. And then I had 3 different species of plants. I had one long-stemmed leafy plant, one grass-like plant, and then I had one short-stemmed leafy plant. I would include their scientific names if I knew them, but I don't have them from the store. Um, I also included one small sample of algae. Methods and materials. For resource gathering, I went to the pond. I grabbed roughly 400 grams of soil below the water level, and then I did 473 milliliters of pond water, and that equates to about 2 cups each. And then I collected one dead leaf and 2 small sticks from the pond, as you can see right there. So week one, we're going on to observations now. Week one, the artificial microcosm. Its water was cloudy from the initial setup, as you can see. You can kind of see it's murky a little bit. Plants were equally spaced out. I tried to have them about like 1/3 apart from each other. So like we had the small leafy plant over here. I'll zoom in so you guys can see a little bit better. I'll probably do the same thing for the week one because the water's a lot clearer for that, or for week two. So you can see the small leafy plant over here, the long stem leafy plants directly behind it, and then the grass is kind of up front towards right here. And then there was a small amount of surface algae from the sample I put in there. So week one for the Hill microcosm, the sediment was completely covering the entire area. I couldn't see anything. The visibility was reduced to 0. There were some root systems inside the the substrate, but that was like when I actually just initially collected it. It was a very rooty dirt when I grabbed it. You saw there was all those reeds next to it. I feel like they came from that. All right, so we're going to move on to week two for the artificial microcosm. I feel this is a better picture to show off the plant life in here, so I'm going to do that. This is the leafy, short stemmed plants I was talking about. These ones, they grow into really long stemmed leafy plants in the back, and then right up here in the front, you can see that their grasses is kind of right there. You can kind of see that stem with the grass. Keep note of where this is placed because a little interesting observation towards the end there. But the water became a lot more clear. We had some small algae growth. The water line's over here, and then we got some condensation on the lid. I'll zoom back in so you can see that. The condensation up here, you can see that. So there's some evaporation going on in there. So at this point, this is where I took my last sample from this because I knew that I was going to be going into doing the hill. So we have week two for the hill microcosm. The water sediment is still pretty bad. You can, sorry, meant to zoom in. You can see the leaf now, at least a little bit, so it's like almost a quarter of the way visible, but the visibility is still really bad. The algae is beginning to collect on the surface. You can kind of see the little specks right there, and over here, that's all algae, and then that's where it's settling at this top. And then I did see some heterotrophs. It was really hard to make out because you see, as you can tell, the visibility is pretty bad. But when they got close to the glass, you can kind of see them swimming around, and they were very small. Like I can't express to you how small they were. Like imagine seeing like a mosquito larva, and that is like 5 times the size of how big these things are. They're just specks basically, and that was basically all I had to report for week two. So moving on to Hill Air Force Base week three. The water became a lot more clear. As you can see, the power panel behind it, you can see straight through it. So the water is like completely clear at this point. And algae begins to appear on the glass itself, not just the surface. So you can see that there's all these little specks. That's all little chunks of algae forming on the glass. So if we go down, you can see some of the sticks and the root structures I was talking about. And then if you look over here, these are air pockets. They're just starting to form in week three. And then you can't really see them in this photo. It's not very well, it's very hard to tell. But there is grass shoots shooting up. I didn't even know that they were grass shoots until week four or when you can actually see them a little bit better. But they were starting to form in this week. I just didn't know what they were. You can kind of tell that there's condensation up here too. I didn't really talk about that in last week, but you can kind of tell that there's a little bit of condensation up here as well. Inside the lid. It's a lot harder to see just because of the mason jar and how it's set up, but the water line's like right here. So you can kind of tell that there's condensation up there as well. Okay, week four. This was the biggest jump in time from my observations. This one I think was 9 days apart, just because of how the observations fell in the week. But this clearly has the most substantial difference between the weeks. Like you can see the algae not only is completely all around it, it almost gives the water like a green tint. I actually thought the water itself was green at first, but I later determined that it wasn't for reasons I'll get into later. The water, the actual algae is starting to go down the water column now. It's not just staying at the surface as well throughout the middle of it. It's not just collecting on the sides. As you can see, these grasses really shot up. I think they look pretty neat. I like that one's kind of bent over a little bit. And then you can see a lot more air bubbles in the system. More air bubbles over here. And this week was the week that we encountered our first worm. So I did encounter a C1 white worm. I don't have a picture of it right here, but I do have later in the um, presentation. But one thing to note is that there is a lot more heterotrophs in here. Your invertebrates I was talking about earlier, I think there's about like 20 that I could count right now, and you can't really see them in here. Like they're just so small. I can't express to you how small they are. Like, and if you, unless they're moving, you really can't see them. Okay, so moving on to week 5. So I actually did take a week 5 artificial microcosm because this is the week we did our forum posts, and I wanted to just have. I wasn't sure if I was going to use it. I don't think I actually ended up using it in the week 5 forum post, but I did take an observation nonetheless, so I decided to include it here. So as you can see, that the algae is starting to cover most of the structures, including the plants. The leafy plant, especially, like half of it had algae on it at this point, and the grasses kind of grew through it. Um, Not a lot of growth from the stemmed plant, but there is a lot of algae on the side of the tank now. So, this kind of there's more condensation in the lid, like I was saying earlier, and then now there's beginning to become air pockets on the bottom of the substrate. If you can see right down here, there's some air pockets down there. And no snatch tail eggs, the snail eggs, unfortunately. In fact, at this point, the algae is starting to cover that rock. But if you look at the rock, you can actually see those white specks. Those are all the snail eggs. So we're going to move on to week 5 for the Hill Air Force Base microcosm. So the algae throughout the water column, it almost looks like a web. So algae is all the way up and down now. It's even touching that leaf. You can see how thick it is right there. And then it's completely all over the side of the glass to the fact that it like makes the water look green. We got some more growth on this for the grasses, and then even more air bubbles. Like the air bubbles are so advanced at this point, there's pockets that look like it's actual just water, but it's really just that's like a whole air pocket. So like the air is really filling that up as well. At this point, the invertebrates became too hard to count. There's probably around 30 in there. I really don't know, honestly, because they move around and visibility is a lot worse now than it was just due to all the algae. I did find 2 more worms, so there's a total of 3 worms for sure counted in here. And then I think that's it for this week. Yeah, that's the majority of it. I have some other observations that'll be later, but I'll talk about those in a bit. Um, So lastly, we have our week 7 artificial microcosm. So the algae has taken over the tank pretty much, and then there's no snails. No snails hatched, unfortunately. Big sad. The air pockets in the substrate as you can see, they're still down there. I didn't really notice a big change in them. They kind of look the same from the last time I looked at it. And then this is something I found interesting is that remember how the grass was in the middle when I planted initially? Well, it grew out towards the side of the wall. The other plants didn't do this, and the small leafy plant, it got bigger, but it didn't grow towards the center. It just kind of grew out in all directions. And the stemmed plant, it really didn't grow at all. So this is the side that was facing the light, though, that I had in there to keep in mind. So one thing that I realized why the water wasn't actually green is because since this lid you can see straight down. This is also taken from week 7, by the way, but it you can see straight down into the substrate. So if you look here, you can see that there's algae over here, but this corner right here, you can see all the way down into the black substrate, and that's how I knew that the water itself wasn't green, but it was just the algae coloring the glass that made it appear green. But the water was actually green. It wasn't actually green, it was clear. So I got a couple videos for you guys, just to show you guys what the heterotrophs inside there look like, the worm and the invertebrates I was seeing. We'll do the one on the right first. So if you So you can kind of see him bobbing up and down there. There's one in the back too. It's only 6 seconds long. We'll play it again. But you can see the one in the back. He's kind of moving around. And the only reason this one looks so big is because it's right next to the glass. This one's like in the middle of the tank, so it's really hard to tell. And then over here, where the circle is, this is where the worm is. So I'll play this one now. You can kind of see him moving. I'm not going to put my cursor over there so you guys can see. But it's like right dead center of the circle. You can kind of see him move around there. So that's one of the worms I was talking about. Okay. Moving on. So I did a couple comparisons of just the two different microcosms and how that both are affected. So if there's one thing that I take away the most out of it is that I really like the cylinder jar a lot better than the mason jar. Just the bigger volume gives you a better chance to see, and then the fact that you can look down at it is really cool as well. Like I would have never known that the water wasn't actually green had I not had this artificial one, because I would have just assumed the water was green because that's what I was able to see. So it gave me a different outlook and different perspective on the observations that I didn't get. So something that might not be as big of a deal for the Hill Air Force Base as if it has that top-down view because the algae was completely up to the top and that's where it started forming. So I understand that it might not be as big of a difference, but I would still like to have that observational view just because like there might be air bubbles or something on there that photosynthesis have been generating inside that algae that I might be able to observe, but I can't like just because it's got a solid lid on it. So that's one thing to keep in mind. So there's substantial growth in the hill microcosm compared to the artificial microcosm. Like, yes, the plants grew, and yes, you can see there's a lot of algae, but like, look at how much open space is on this compared to this. So it might be because the light it is closer to the light source, but like I said, they're kind of separate. They're it's not that much farther back. It's like maybe what 8.6 centimeters back further than the other one. And not only that, but this had 7 whole weeks to gestate, where this one only had 5, and the growth I feel like is still a lot bigger. That might do just because of the volume as well. There's a lot of factors that could play into it, but the Hill Air Force Base did have noticeably more growth as opposed to the artificial one. One theory I have too is that since there were heterotrophs inside the Hill Air Force Base one, and there really wasn't inside the artificial one, there was more CO2 for the plants to grab as well, just because they have more things respirating and they're not just relying on their own respiration from the plants themselves. So there would have been more dissolved CO2 in this ecosystem. So I think that might have had something to play with it as well. Okay, so we're going to move on to interesting observations. So one thing I found that was really interesting is that this is a completely isolated section of algae, and it is in the substrate of the Hill Air Force Base microcosm. So it's like a little circle. It doesn't go anywhere else. It just stays right here. This was the side that was facing the light, so I understand that it was under higher light conditions than the other rest of it. But even up towards here, there's no algae. It's just strictly at the bottom. So I'm really curious as to why that happened. It's got its own little microhabitat in there. It's pretty neat. And then something else I found really fun in this red circle, you can't, let me zoom in a little bit. Maybe you can see a little bit better. It's kind of a glare, but this grass is actually growing upside down. So one thing I think is since it's on the actual side of um the jar, it doesn't really care because it's getting its nutrients from the water anyways, and it's still getting light. So I don't think it knew to self-orientate it to go straight up just because it was already receiving light from the side of the jar. So I thought that was pretty interesting. So we have a quick little summary of the data tables for Hill Air Force Base microcosm. So we have it sampled by week, watercolor clarity, substrate changes, autotrophs, and heterotrophs. So for week 1, we had brown, not clear, large amounts of sediment in the water. And then 2, we had beige opaque. And then we had 3, it was clear. And then again, like I said, I thought it was green, but it really turned out to be clear. Like I just thought that because of the algae. So like I thought it was green, but it was actually clear for those 2 weeks for week 4 and 5. We had subset changes. There was no change for week 1, and then week 2 there was no change. We had bubbles starting to form in week 3, and then more air bubbles, and then we started to get some tunnels from the worms in week 5, and we had even more bubbles for week 5 as well. So autotrophs, we had algae and roots for week 2, algae roots and grasses for week 3, algae roots and grasses, algae roots and grasses for week 5. Heterotrophs, we didn't see anything. First week, there's no visibility. We saw about 12 swimming around in week 2. Didn't see any more in week 3, and then in week 4 we had about 20. And we saw that first white worm, and then week 5 we had about, I honestly couldn't count, but I'd say around be 20 to 20, about 25 to 30 probably, and then we saw those 3 white worms. So observations and data summary table for artificial microcosm. We did not have any data for weeks 3, 4, or 6, but we did do it for 1, 2, and 1, 2, 5, and 7. So I'll go over those. Initially, the water was cloudy, then it became clear, and it stayed clear for the rest of the time. There was no change in substrate for the first week. Bubbles started to form in week 2, and then lots of bubbles in the substrate. And then I put no changes for week 7 because I didn't notice any more bubbles. Autotrophs. We had the long leafy plant, the short leafy plant, the grass, and the algae, and that never changed. Obviously, there was growth within them, but as expected from an artificial created environment, there was no surprising plant materials that came up. And our snail eggs for our heterotrophs, unfortunately, they did not hatch, so I just left them there, and then until you can determine that they're dead, I just left them on the heterotroph side. So major findings. So this microcosm showed that there is abundance of life in the Hill Air Force Base duck pond. The fact that it completely disproved my theory that there'd be no heterotrophs for my hypothesis, and then the cold water had, for the invertebrates, they had to have been in there before they weren't like dormant or anything, because like the minute I was able to see into the microcosm, you could see that they were swimming around. The worms I'm not so certain about, just because I didn't see them till like week 4, so they could have been dormant and they kind of snapped out of it due to the warmer waters, but I'm not entirely positive about that. That could go either way. And then we had a prominent amount of algae in both the systems, so due to the high light factor, like I predicted, so both hypotheses were confirmed for that. And our snails didn't hatch. I didn't write that down here, but that's something that I already talked about as well. But our snails didn't hatch, so I'm assuming the tank was just too cold to actually get them to incubate. So we had and then our cylinder latching jar is a by far superior habitat, due to be able to see from the top down, and you have better viewing overall. Okay, so conclusions. So my key concept 1, I used energy levels. So in order to sustain a balanced ecosystem, there must never be more primary consumers than there are producers. And then microcosms put this in full display. So roughly 10% of the total number of producers will be primary consumer. Roughly 10% of the total. Sorry about that. I read that really weird. But for the amount of producers you have, 10% of that will be primary consumers, and then another 10% of that will be secondary consumers. So if there was 1,000 producers, there would only be 10 primary consumers and 1 secondary consumer. This microcosm, I feel like had actually pretty good display of that. As you guys saw, the actual algae and other producers just took off. There was a ton of them, and then after that, like they started growing. So did the primary consumers, which I'm assuming were those vertebrates. I mean, invertebrates, the little small shrimp-like bug things. They took off too. So I feel like that this was a really good display of that. Those worms, they might be secondary consumers. I'm not entirely sure. I'm pretty sure they might even be decomposers. I just don't know. But they also kind of fit into that line where there really wasn't a whole lot of worms compared to everything else. So if they were secondary consumers, it would have lined up with this. So I thought my microcosm did a pretty good example of showing this. So something my microcosm doesn't do is show a predator-prey dynamic. By creating a microcosm with first and secondary level consumers, you can study prey dynamics by showing how micropredators feed on other organisms such as like leeches or other carnivorous animals. You can kind of create a population prey species and actually show those S-wave dynamic charts about how prey populations go up and predator populations go down. I think that's something that microcosms are really good for doing on a small scale. Obviously, you're not going to be having rabbits or anything like that in here, but like from microbial life, I think it's a really good way to observe that. And then key concept 3, I chose biogeological cycles. So oxygen and carbon cycles are clearly displayed in microcosms. Photosynthesis creates oxygen for the animals to respirate and create CO2 and then continue plant growth. The water cycle continues through the jar as vapor condenses on the glass and then returns back to the system. So there's a lot that you can get into for biogeochemical cycles. Like just for carbonate as an example, like as the plants use it to they not only use it to respirate, but they also use carbon to produce sugars. So as they're respirating, they're breaking down those sugars as well. Not only that, but the heterotrophs are eating the algae and other things that so they're using the carbon and breaking down themselves, and eventually they'll die, and then they'll decompose and go back into the system, and it just continually cycles over itself. That's just like one example. I really don't want to get too in the weeds because you can go really far into biochemical cycles with microcosms just because they display so many. Like I said, the water cycle goes through there, and respiration is part of oxygen as well. So it just kind of keeps looping back on itself. So one thing that I think microcosms can really be useful in ecology is using them to actually take an environment and then subjecting it to some sort of toxin or controlled environmental change. So that was actually something that the artificial microcosm really taught me is just because I had that heat pad on it. It was like you could just take an ecosystem and then just subject it to whatever you want to in a small enclosed real time access. So say you want to test global warming on how affecting the temperatures will on this environment for a prolonged period of time. You can do that. That's something that microcosms allow. You can just pull that chunk from the environment, take it, put it in a jar, and then subject and then be like, hey, you're going to be at 95 for 7 weeks or however long you want to test it for, just to see how the ecosystem handles it, like if it collapses and whatnot. You could also do this with heavy metals, toxins, mercury, stuff like that. Be like, hey, how does the soil affect this? Does it absorb it? What do the plants absorb it? How do the microbial life in it? Do they can maintain it in their bodies? Is this what builds up? Will it stay in them? There are so many things that this can answer just from testing these one things. So like pollution effects understanding is something I don't understand why I haven't seen more studies based on that. I think that's something that could be really beneficial in the future. And then also, it allows ecologists to view extremely small creatures that would be very difficult to do in the wild. If I would have done this just from walking out to the pond and looking into it, I would have never saw any of those invertebrates. And as you saw, there was 12 of them just in that little jar from when I started. So these allow ecologists just to take a little chunk of the river or whatever they're doing, put it on a shelf, and then they can look at all the microbial and small insect life or whatever else is going on there in the comfort of a lab where you just wouldn't be able to make those observations in the wild. So that's something I think is also very beneficial from microcosms. So for a future research, something that so what further hypothesis would you like to test, and then what would you do next time you want to improve the project? So something that how small those invertebrates were actually makes me want to get a microscope and look at them through the microscope just so I could see what they look like because they're just so small. It's really hard to get a really good look at them. I would also take more detailed observations of the artificial microcosm and add an invertebrate, something like an actual snail or like a crustacean, like a cherry shrimp or something, just to see if that would help keep down the algae and under control better. I would also like to test the Hill microcosm using the cylinder jar just to see if I get a better view from all sides, just because it had a bigger volume and I don't want to look down from the top of it as well. And then I want to know why the bubbles form in the substrate. I want to say it's related to the nitrogen cycle, but I don't know. I need to do some more research onto that. So those are some things I want to look into moving forward. And then these were my references. Okay, and that was my microcosm project. Thank you for listening.